And dear audience, please help me now to congratulate Kavli Prize laureate and geneticist Huda Zoghbi. All right, there you are.
Thank you.
Huda, you are based in Texas, US, at the, among other institutions, Texas Children's Hospital. And your work has helped reveal genes and genetic mechanisms responsible for a number of devastating neurological disorders, such as one we might know all about, or know of, the Rett Syndrome. But when you started your work, Huda, your education, you had to escape a civil war in Lebanon, and this was back in 1976. How did that influence your education?
It did influence my education. It wasn't planned, I thought life was perfect in 1975 when I started at the American University of Beirut. Beirut was the most vibrant city, Paris of the Middle East. Nobody dreamed of its going through a war, but when that happened, and after a very rough year, the first year of the Civil War, my parents go to the States for just a few months. You'll come back in October, and hopefully war will be over. But many of us know wars can take a long time, and it wasn't over, and I had to find a school that will accept me in the United States and transfer. And could not return for quite a few years, and that's why I'm still there.
Please, the floor is yours now,
Thank
Huda.
you. Thank you so much. So it's really a pleasure to be with you here today. I totally agree, you are the future, and I would say perhaps this is the most important event I will do while celebrating the Kavli Week in Norway. So thank you for being here. And I'm gonna give you one more challenge in addition to your Instagram challenge. I hope I'll get many questions, and I would go back to the United States, and I will say some of the most interesting questions came to me from high school students in Norway. So I encourage you, that's the challenge. So as you've heard, I stayed in the US and finished medical school there and decided to do pediatrics. I was on a path to become actually a pediatric cardiologist. That was the reason I went to Baylor College of Medicine when I did my residency training. But as I rotated in pediatrics, you do different rotations. As I rotated in different specialties, I was fascinated by my pediatric neurology rotation, where I discovered that by talking to the patient or to their parents, because these are children, just by piecing together all the symptoms, you can tell where the problem, which part of the brain is the problem coming from. And I found that fascinating. To me, I love solving puzzles, and to me, that was like solving a puzzle. And although everybody was expecting me to be a pediatric cardiologist, the cardiology department was saving a fellowship place for me, I switched in my third year of residency, choosing to do pediatric neurology, which is really, Mr. Secretary, to follow your point, follow your heart. I could not extract myself from the joy of solving problems. So I did pediatric neurology, and quickly, this was 1982, now when I started my residency, I quickly discovered this is a really hard specialty. It's one thing to be fascinated by a problem and to try to see which part of the brain it's affected, but it's another to watch a child suffer with a neurological disease, to watch a parent cry when they know their child has a degenerative problem, they will die, and this problem may happen again, maybe 1 in 2 or maybe 1 in 4, depending if it's dominant or recessive in their family, but we did not know the cause of. We couldn't tell them what caused it, we couldn't tell them how to prevent it. And it was about that time that I encountered a young child who was perfectly healthy, and by 2 years of age, lost the ability to sing, nursery rhymes, to say few words, couldn't anymore use her hands, constantly wringing her hands, and became, within few years, imbalanced, lost language, autism, seizures, devastating. So I'm looking at this child and thinking how painful it is to have a beautiful girl and then for that girl to lose all these skills, and how painful it is for a parent to see this. And just by chance, a week later, I saw another girl. And it happened that that same month, a paper from Sweden, very close by Bengt Hagbert, has reported on European girls, 35 girls with Rett syndrome. So think about the coincidences, right? Truly serendipity. A paper comes out in October 1983. The same week I see a patient with Rett syndrome. And a week later, by chance, I see another girl. She came with a diagnosis of cerebral palsy who had Rett syndrome. And I found that very painful. That together with all the really heart-wrenching cases of child neurology I saw in the clinic, that's when I decided I can't do this anymore. I have to do something. And the only way I could think of doing something is to finish my clinical training, leave it for a while, get into the lab, learn how to do research, learn how to do molecular biology and genetics. Maybe I will find the causes of these diseases, and maybe we can do something about them. So that was my path. You will hear most scientists go for science just because they're curious about something. They're not connected to any particular problem, they're just curious. But in my case, I said, I want to find the cause of Rett syndrome. It intrigued me because the girls were born normal. They lost all their skills, but it's not degeneration. So how come the brain is still all together there, but it's not working? And I'll tell you later what we learned. So that was the reason I went to the lab. It wasn't easy. I could have made a lot more money, immediately becoming a professor in neurology, making money, seeing patients. But I went back to a very low-level fellow in a research lab who knew less than the graduate student, because the graduate student had a recent college education. They knew the latest in molecular biology. Mine was many years before. So it wasn't an easy experience to be the least accomplished in a lab after you've done so much, 10 years of medical school, pediatrics, pediatric neurology. But for me, I felt it was important, and I'm very glad I did that. I will share with you one secret. Anytime you start something new, it's really hard. And going back from being a very well-established clinical neurologist to being the one who knows the least about any experiments in the lab is very hard and humbling. And it's easy to immediately see failure, because when we do experiments, we don't always get positive results. It's easy to experience failure and to immediately say, I'm going to go back to the clinic. I'm accomplished, I can do this, I can be successful. It's easy. But I did promise myself one thing. I'm going to give research 5 years. I will see at the end of 5 years if this is for me or not. No matter how hard it is, no matter how many negative data I will get, it's getting 5 years of me. And that's what really happened. I think if I have not done that, I would have quit half the way. So my advice to you, when you start something new, give it a chance. Maybe 5 years is the right time, maybe 4, but give it a chance. Don't give up on something very quickly. And I've learned that lesson all my life, and now I take life 5 years at a time, because things might change. You cannot take it 10 years. So many new things will happen. So in the lab, I wanted to clone the gene for Rett syndrome. I had to find a mentor, and I had a wonderful mentor who was the most famous geneticist in Texas, not only the city of Houston. He writes actually the book that you might study, The Molecular Basis of Inherited Diseases. And he told me, what do you mean you want to work on Rett syndrome? Show me the families. I said, there are no families. It's just one girl here, one girl there. It's a disease that just randomly happened. And he looked at me and he said, you cannot study a disease that randomly happened. This is a genetics project. This is a genetics lab. You have to study a disease that's Mendelian, that it's inherited, either recessive or dominant. There's no way you'll ever find the gene for a disease that's just one case here, one case there, random cases in families. So that was hard, and I had to face the decision, either leave his lab and find someone else that will take me, or listen to him. And knowing how great he was, I listened and decided to find a Mendelian problem, picked a genetic disease that's extremely rare, 1 in 100,000 people. It affects balance and coordination, but I picked it because that's where I was curious. I was curious, why is this dominant disease causing symptoms? A dominant disease is when a gene comes from one parent and you have a healthy gene, so one mutant gene and one healthy gene, and you have that. Why is it that the healthy gene doesn't protect? That's one curiosity. The second curiosity, this is a late onset degenerative disease. Why is it that those people are born with a mutation, but it takes 30 or 40 years for them to develop balance problem, swallowing problem, and they'll eventually die within 10 years. But for 30 to 40 years, they're fine. And I thought that was really interesting, and that intrigued me. So I started working, I found a big family, started working on that disease, and we discovered the gene. I collaborated with Harry Orr, who's one of the Kavli Prize winners this year. We identified the genes together on the same day. It was a beautiful collaboration. That was really chance, but we found it exactly on the same day. We were sending each other faxes. There was no email at that time. Or there was email, but we're not using it much for data. So we sent faxes of our pictures, and we found the gene together on the same day, and we started working to figure out the mechanism of that disease. And I'm happy to report to you that today, thanks to all that work, we were able to today bring therapies to that particular disorder. There are 2 to 3 companies who are working with us now to try ways to treat that disease. But I had not forgotten Rett syndrome. That was my passion. That's why I went into the research. So I kept trying to think, how can I narrow it? It's literally like trying to find, one of you, trying to find a person in the United States. You don't know their address. You don't have a map of the United States. You don't know what city or state they live in. That was the situation, trying to find the Rett syndrome gene. Genome was not mapped, and I had no idea where to look. But started using logic, thinking since all the patients were girls, maybe it's on the X chromosome. Maybe I'll focus on the X chromosome. And focusing on the X chromosome, starting sequencing genes from the X. We did sequencing in the bench at the time. I then got lucky and found 2 families where there were 2 sisters affected, but the parents are healthy. So if this was on the X, a mom could be healthy because of X inactivation, where most of her cell express the healthy allele. And that helped me narrow down the region to 1/3 of the X chromosome. And then another family, a few years later, helped us narrow it down to about 10 million base pairs. And from there on, it was just brute force. Sequence gene by gene, till we found the gene in 1999. So from 1983, when I first saw Ashley, my first Rett syndrome patient, till we found the gene, it was 16 years. And the message in that is that if you really believe in something in your heart, you should follow it. But don't be foolish. Don't make it the sole thing you do, because it's pretty risky. That's where my project on the ataxia was helpful and kept my lab running. I could train students, I could get grants, but at least I believe that Rett is going to be a gene when no one else really believed. Even funding agencies did not think it will be a gene. But I believe, and people ask me, what is it that made you believe that? I say it was the girls. Every girl I saw looked like the other. Born healthy, by 1 to 2 years of age, lose language, lose hand use, wring their hand, and have all these other problems. When we found that gene, it turns out to be a gene. When we studied it, we made mouse models. It's really important for all aspects of brain function. All of you sitting here today listening to me, the reason you're going to remember this conversation is because this gene is helping few neurons that in the center of learning and memory to be active together to retain some facts, I may say. You're not going to remember everything I say, but something might resonate and make you remember it. It's going to coordinate which of these neurons are capturing the information and retaining it. If you don't have that gene, you can't do that. If you sit down to eat, your body will sense, your neurons will sense how much food you have and will tell you when to stop. If you don't have that gene, you will eat uncontrollably. If you want to move your arms, how much force to put, all these neurons are trained by this gene to know how much to do, how to plan a movement. So it really turns out that it affects all part of the brain, and that's why girls with Rett syndrome have so many problems. We also learned that it's a gene that's a Goldilocks protein. Now, Goldilocks, that's an American story. I don't know how many of you have heard about Goldilocks and the three bears. You've heard it, good. So it has to be just right, not too big, not too small. This gene, you have to have it, we learned from studying animal models, it has to be just right. Not too little, you get Rett syndrome. We learned if you have twice as much, you get another neurologic disease that's devastating, autism, intellectual disability, terrible seizures, and actually that one shows up in boys and causes death by 20 years of age. So we've got and we learned that from making a mouse that had an extra copy. It was a curiosity experiment, and the mouse was very bad, and we predicted it'll cause a human disease. And Linda Van Esch from Belgium actually happened to have patients who had duplications of the Rett syndrome gene. It's called MECP2, for methylcytosine binding protein 2. So that told us that you have to get it just right. And having the animal model for the duplication, we were able to use a strategy to test if we can treat this disease. And that is the latest in neuroscience and therapeutics. You make a very small piece of DNA. It's called antisense oligonucleotide. You know, the DNA makes RNA, and the antisense binds the RNA. So it's a piece of DNA that binds the RNA. In our cells, there are enzymes that don't like to see RNA binding DNA. That's abnormal. DNA should stay with DNA, and RNA should be separate. But when you artificially give this little piece as a therapy, that RNA will be degraded by an enzyme called RNase H. And this way we can lower the protein. Because we degrade the RNA, now we make less. And what we showed is that even after all the symptoms develop, if we do that, we can reverse the symptom of that disorder. And this month, we're having a meeting in Houston with all the families, the pharmaceutical company. We've been doing clinical trial readiness, how to prepare for clinical trials so that they can do a clinical trials for the patients to help those with the duplications. We're still working on therapeutics for Rett. We're not quite there yet. But we also learned that sometimes the therapy may not be a pill or a DNA molecule. Sometimes it could be, we studied the brain network and we identified what's wrong with the brain and Rett syndrome. It functions. Remember, I said there's no degeneration, but it functions at decreased activity. It's reduced by 30%. So basically, if we can stimulate certain areas of the brain, we can correct the symptoms. And there's now techniques that are used in humans by neurosurgeons to stimulate specific brain circuits, and we're working with that. So you could see how from just the observation of one patient, not only did we really come back to find the causes of these diseases, to help the families with early diagnosis, and for the ataxia with genetic counseling so they can have family planning and the disease can stop in these families. But it's also taught us about the function of a molecule in the brain, controlling so many normal functions we live every day. It's helping us understand brain circuits which ones are for learning and memory that you can manipulate, for anxiety, for motor planning, and for balance that we can manipulate. And this has implications for other diseases, of course, beyond Rett syndrome and the ataxia. Now, when I first left clinical medicine, many of my clinical colleagues and friends would say, but don't you love medicine? Don't you love seeing the patients? And I did clinical medicine for 20% of my time throughout all this research career. Why did you leave it for research? Don't you love your patients? And I said, I went for research because I love my patients. I knew this is the only way I could really help my patient, by doing research and being able to bring something back to them. And it took a while for me to really realize that that's a true statement, but I think throughout these years, I now learned that. That was the best thing I could give back to my patient, by understanding their disease, their mechanisms, offering proper genetic counseling, offering potential candidates for therapies, and helping them in other ways by them being part of a community. So this is something I wanted to share with you. The reason now I want to get back why I'm very excited to be with you. Many people think that a discovery was hot, the ataxia discovery was exciting. Some other discoveries we've made in the lab, I don't have to tell you about all of them, were really very exciting, and they were. So people always ask me, what do you consider your most important contribution? What's the most important thing you've ever done in your career? And any guess? Don't be shy. If anybody wants to guess, raise your hand. Okay. Obviously, it's a hard question, because if people ask me that, very few people know the answer, and I will tell you the answer. The most important thing I've ever done in my career is training the graduate students and the postdoctoral fellows and the undergraduate and rarely some high school students in my lab. There have been hundreds of people who've come through the lab, and there have been over 120 PhD and fellows that have come through the lab, and I consider that my most important accomplishment. And there are many reasons why, and I hope you will remember that. I am one person. I will come through this life, make some contributions, be honored, which I'm deeply appreciative of, but I am one person. I can only do so much. But if I take time and help those who are young, and I train people, imagine now this one person, I'm already multiplied 120 by the real fellows and students who've come through the lab and who've pursued research careers. And if each one of those has the same philosophy and train not even 100, but even 10 or 20 people, we're already 1,000 people contributing to science and biomedical research. So I think this is really what I consider my most important accomplishment and what I am most proud of. And last but not least, you heard about a career in science, and is it for everybody? I think it was the career I loved, and I'll tell you why I loved it, for a few reasons. I'm someone who has to be constantly stimulated, constantly something new. I can easily get bored. And science, I'm never bored. Every day I go to the lab, there's something new. I don't know what I'm going to learn today. Positive, negative, it's that unknown. Our brains like the unknown. That's how we get the reward mechanisms. And it doesn't have to be everyday reward. In fact, reward is most appreciated in the brain when it happens so sporadically. And that's what science gives you. And second, it's really a most noble career. You're doing something to help humanity in some shape or form, even if you're giving one building block, even if you're not making the big discovery, because all of our discoveries are building on these other buildings blocks, no matter how small or how big. But last, you don't have to be in the lab to be contributing to science. Science careers have expanded. You could be a writer, you could be in policy, help politicians really make great decisions. You could be in patent law. You could be in science communication. You could work at organizations that fund science. So ethics of science, there's so many things you could be doing with a science career. So I hope my very brief introduction to my career in science gets as many of you as possible. We need you into that field. Thank you very much.
Thank you so much, Huda. This is very, very inspiring to listen to. And I guess when a neuroscientist says that our brain likes the unknown, it must be true, especially when she won the Kavli Prize as well. So let's challenge ourselves. And questions for Huda? Can I see a hand? Yes, in the middle of the row. Please wait for the microphone before you ask your question. And would you be so kind and say your name?
Hello, my name is Nicholas, and my question is, how did you find the gene? Because it's not something that you just see.
Right. So the way we found the gene, as I mentioned, in those 10 million base pairs, there are hundreds of genes. And we started systematically sequencing genes one by one, taking the DNA from the patients and the parents. Parents are healthy. The patient has Rett. And what we did, we find DNA changes in the patient that were not in her parents. And we start finding mutation. We will do 6 girls at a time. We found 6 girls with Rett having mutations that create a stop codon, which means you truncate the protein, you don't make a protein anymore, or change important amino acids, and that's when we knew it's the right gene.
Thank you. Anyone else that would like? Yeah, in the row in front of you. Yeah, lovely.
Hello, my name is Victor, and I have a question about the ethical dilemmas when it comes to researching the different genes, and how prevalent were these when researching the gene that you were most interested in, and how the discourse was at the time in pre-2000.
Right. So before 2000, before the genome was sequenced, we had to do the discovery the long and hard way, where any gene we want to study, we have to characterize it on the bench, and we have to map which part of it makes the protein, and so on and so forth. Today, the genome is sequenced. When a child walks into the clinic today with a neurological problem in our clinic, we send the blood from that child, or saliva even, to the DNA lab, and they sequence all the coding genes, and within a couple of weeks, we have an answer. So it's very different. And even if it's a new gene, it's really much faster and very different discovery process. So that's wonderful. The challenge is when you find a mutation in a gene that nobody has ever seen before. You're not allowed to call it the disease-causing gene unless you've seen it in more than one person. Because all of us in this room, anytime we're born, we will have new mutations that are not in our parents. And those new mutations are few dozen, and typically don't cause any problem. But sometimes they do. And in a case where you find it in a child with a problem, you cannot blame that gene until you prove causality. And the way you prove causality by studying additional patients that look similar to that one case, and if they have mutation in the same gene, that's how you prove causality. And then you mentioned something on the ethical ramification of gene discovery. I think that the most important thing is to really educate the families about what runs in their family. For example, the disease that's inherited, when a person is an adult and they're not having any symptom, but they want to have children, we have to give them their options, and we have to tell them all of their options, and they make a decision. We tell them, you carry this gene. There's a 50% chance for every pregnancy that your child will have that mutation. You have options. You can choose to use sperm from another person or to do in vitro using surrogate eggs if the mother is affected, or we tell them you can adopt, or we tell them you can have a child but then have a amniocentesis or sampling or the chorionic village do diagnosis and see if it's healthy, and you have to decide if you want to keep the pregnancy or not. Or there is a really good option also we recommend where you do in vitro, you look for the mutation in the cells. You say, of all these embryos in the dish, which embryo has the healthy gene rather than the mutant gene, and you only implant the healthy gene. The advantage of this, you do all the work before the baby is implanted, so there's no need to have therapeutic abortions if the child is affected and the parent don't want to continue the pregnancy. So these are examples of all the things we offer people, and then people make their own decision. The key for us is to inform them of all the options.
Thank you so much, Huda. And we have time. Oh, one question, two questions. Okay, three. You're very popular. So how can we move the microphone the easiest way? Lovely, thanks.
Hello, my name is Boris, and I would just like to know how much large-scale neurological research would cost for the state.
I'm not sure if I understood. Were you interested in the cost?
Yeah.
Of the sequencing?
Of the neurological research on
on
average.
the research.
Oh, good question. So he's asking, how much did it cost to discover, say, the ataxia gene?
Yeah.
I can give you very simple math. Harry and I worked together on that for about, I got trained in arts lab, so I won't count this year, but together we worked for 5 years. We each had one NIH grant, that's $250,000, that's $500,000 a year for 5 years, $2.5 million for us to discover the gene. But that's because we collaborated. I think if we haven't collaborated, it would have either took us longer and obviously cost more money. By us working together, we complemented each other, and we always did the complementary experiments. It took more for Rett syndrome because it took longer to find. Probably, I would say, it probably was close to $4 million for us to find the Rett syndrome gene.
Next question.
Sorry, and today you can find it in $100 because this is how much it costs the exome. And this is what's exciting. I mean, think about me spending 60 years to find something that you can find in one day. Our genome center can sequence a whole genome in 7 hours. Think about that. So this is why it's such an exciting time for you to be doing science.
There was one question there on the outside behind you.
Hello, my name is Horek. I'm wondering, are there any other neurological diseases that you hope the next generation of scientists may discover and hopefully cure?
Great questions. There are thousands of neurological diseases waiting for all of you. So one of the other winners worked on a disease called Fragile X, which was the first neurodevelopmental disorder gene to be discovered. I think Rett may have been the second or the third. And after that, people started looking for these new mutations. And today, there are 1,000 genes, literally 1,000, that cause different forms of neurodevelopmental disorders and autism. That's in the neurodevelopment field. We discovered, Harry and I, the first ataxia gene, which is balance disorder. Today, there are 200 ataxia genes discovered, and they are close to about another 100 to 200 to be discovered, where the gene hasn't been discovered. But then there are Parkinson's, Alzheimer's, the psychiatric diseases waiting for someone like you. There's so much to be discovered. And anything you work on deeply, you're going to reveal beautiful biology. If you picked a random gene, so there's a lot to be done.
I see you have a follow-up question, but there is one before you, in the middle of the
And he
row.
has a question here, too.
Hello, I'm Brage. You talked about a new type of therapy for this neurological disease that is going into clinical trial. How often do the patients need to use this? Is this like something that they have to do the rest of their lives, or is this like one time or something like that?
Excellent question. It's something they're going to have to do sometimes maybe twice a year or maybe 3 times a year. It has to be put as an injection, like a spinal tap, and they have to get the medicine so that it gets through the brain. Eventually, we hope we can develop something that maybe will correct the genetic defect, and it's a one-time deal, but we're not there yet. So we have to use this therapy that you will give frequently until 3 times a year until we have something more definitive. Great question.
Thank you. And the gentleman in front here on the second row.
Okay, women, help me out. Why is it only boys asking questions?
Yeah.
Come on, please.
Yes?
So I was just wondering, you talked about how easy it is to just give up if something gets hard. How do you think your life would have been different if you hadn't continued doing research and instead just kept seeing patients and kept going down the route you were prominent in?
I think that's really a good question. I think it would have been still rewarding being a clinician and helping people as best as you can. That would have been still good. I just don't think it would have been as meaningful to me because I don't like to use Band-Aids, you know? When you just tell the family, sorry, we can only do this, just do some physical therapy, it's not enough for me, right? I wanted a little bit more definitive. So for me, it would have been fine, but this ended up being much more rewarding.
We found 2 ladies here. And the gentleman that already had a question, you kindly wait, so we get the mixture a little bit. And we are going over time now, ladies and gentlemen, because you are so interested. And I hope we will have astrophysics in a few seconds. Yeah.
Hi, my name is Katja, and I would like to question, like you have said that you use like 60 years for this research. If you were given like extra more 60 years, what would you like to research? Like something new?
So,
future plans?
What am I doing next?
Yeah.
Okay, so now that we have these genes, we learned very much from both disorders. There are a couple of things I'm doing. I'm studying new ways to treat Rett syndrome. Remember, we only now have a therapy for the duplication. So my next 5 years are focused on finding new ways, and we're working on that to treat Rett syndrome. That's one area of research. The disease with neurodegeneration, although we're moving to clinical trials, we're still curious about in many neurodegenerative diseases, whether it's ataxia, Alzheimer, Parkinson, the genes are expressed everywhere, but only 1 or 2 parts of the brain degenerate. We're doing research to understand why. And last but not least, we're coming with new strategy to figure out how to prevent the dementia that you see in Alzheimer's disease. So we're starting to find new ways to prevent toxicity from these bad proteins that drive Alzheimer's. So this is my future areas.
And further on the lady in red.
My name is Mari, and I wonder, you said you have to give science some time, 5 years, and what made you sure that you wanted to move on with science after that?
Great question. The question is, I said I'll give it 5 years. What made me stay? And a couple of things. The first 3 years were all negative data. Thank God I decided 5 and not 3, because I would have detoured. But after those 3 years, I started getting closer to the ataxia gene, and I wrote a government grant, we call them R01, and I got it. And that's another 5 years of funding. So I sort of felt, okay, I'm inching my way closer to finding the ataxia gene, making progress, and I have now this 5-year new grant. I'll stick with it. And then, you know, the rest is history. You become so immersed, so many discoveries, you stick with it.
Thank you. And the final question is up here next to the camera.
Hey, you said most people didn't believe you when you told them that Tourette's was caused by genetic mutation. What other hypothesis was there for the origin of this now known neurological disease?
So it's Rett, not Tourette. Those are different disorders.
Yeah.
Rett, right. So yes, people did not think it's genetic because it was random cases. But now we know, you remember those 1,000 genes I talked about? 99% of them are like Rett, new mutation. So that became the norm. As more and more discoveries, that became the norm. That this is what happens in autism, intellectual disabilities. They are really new mutations.
Yeah, but for the people who thought that this wasn't a genetic disease, what other hypothesis was there for origin?
Oh, they thought maybe not genetic, perhaps. Maybe environmental.
Oh.
That was their other hypothesis. Yes.
Okay.
So for those of you, I know many of you had other questions, and many women, I'm sure, had tons of questions. And you go to Google, find my name, email me. I will answer you. Thank you so much.
Thank you, Huda. Wow.